much. And I'll pass it now to my
Thank
colleague Alexis. He's going to moderate the next presentation. Thank you.
Thank you, Amir. Now we have Veronica Lisi from University of Roma with a talk entitled Exercise Stimulated Extracellular Vesicles Promote Cardiomyocyte Pro-Survival Programming in a Redox-Dependent Manner. Hi, Veronica.
Hi.
It's a pleasure to have you here.
Can you hear me?
Yeah.
Okay, perfect.
Feel free to share your presentation attached.
Okay. Thank you for this possibility, and welcome everybody. I'm Veronica Lisi, and I had my PhD in human movement and sports science the last of May. And the aim of this poster is to show some data of my PhD project that focus on the protective role of EV released before and after a single endurance exercise in a human cardiomyocyte-induced model. So, as is good known now, in regular exercise is crucial for the prevention of different chronic disease, as well as cardiovascular disease, in particular because regular exercise can stimulate redox homeostasis, and this balance can promote the antioxidant response and the counteractive of ROS, the reactive oxygen species. And this is important because reactive oxygen species can contribute to the initiation and to the development of cardiovascular disease. And so this adaptation, this mechanism of adaptation to redox homeostasis mediated by physical exercise or better regular exercise can be mediated by intertissue communication. And in particular, in the last years, some authors see how physical exercise can induce the release of extracellular vesicles in circulation, and in particular, physical exercise can modulate the cargo of these extracellular vesicles. But since the role of this extracellular vesicle released during a physical exercise, and if they have a protective role, is not really really understood, we decide to study if the EV released during a physical exercise can have the protective role on human cardiomyocyte. So I want to better explain you materials and methods. So we enroll a total of 21 healthy young male, and they perform 30 minutes of endurance exercise on a treadmill at 70% of heart rate. And we collect the blood before and immediately after this exercise. And we isolated the extracellular vesicle from the plasma with the site exclusion chromatography, and we characterize with the Western blot for the canonical extracellular vesicle biomarker. And after, we decide to use this extracellular vesicle to treat for 1 hour an induced model of human cardiomyocytes. Okay, let's go to the first result part. In the first part, we decided to study in deep the extracellular vesicles released before and after exercise. And we can see that just 30 minutes of endurance exercise can modulate the total number of extracellular vesicles. Indeed, we have a decrease after the exercise, and but these extracellular vesicles increase in size. Anyway, what is very important that we saw an increase in antioxidant enzyme catalase, such as catalase and glutathione peroxidases in extracellular vesicles released after physical exercise. And moreover, we measure the concentration of reactive oxygen species with the GCF probe, and we saw that these extracellular vesicle, the extracellular vesicle released after exercise, show a decrease also in reactive oxygen species concentration. But the next question was, what happened when we treat for 1 hour with this extracellular vesicle, an induced model of human cardiomyocyte? We can see from this plot that the cells treated with the EV post have an increase in antioxidant enzyme activity, such as glutathione peroxidase, catalase, glutathione reductase. Sorry, here there is a mistake. And there is an increase in the antioxidant activity respect the cell treated with the EV released before the exercise, and the cell treated, not treated. Moreover, we measure an oxidative stress biomarker with the T-bars that indicate the lipid peroxidation, and we can see that the lipid peroxidation is decrease in a cell treated with the EV post, and moreover there is a decrease also in reactive oxygen species in this cell, measured with the DHA probe. Well, the last part, and for me, I think is the most interesting, is that 1 hour of post-EV treatment increased the nuclear erythroid factor 2 translocation. The nuclear erythroid factor 2 translocation, NRF2, is crucial for the expression of the antioxidant enzyme such as catalase, superoxidase mutases, and hemioxygenases. Indeed, in immunostaining analysis show an increase of the nuclear, the NRF2 translocation in cell treated with the V-post, but after 6 hours, we saw also a significant increase in the expression of SOD2 and hemioxygenases in cell treated with the post-EV. So we can conclude that 30 minutes of endurance exercise can change the number, the size, but in particular the antioxidant enzyme activity in plasma extracellular vesicles, and this can impact the redox homeostasis balance of human cardiomyocyte exposed in vitro. In particular, our results show that there is a direct translocation of the extracellular vesicles cargo in these human cardiomyocytes. And that's it. Thank you.
Thank you so much, Veronica. It was a fast presentation of very interesting results, and I think that many of the members of our audience will be interested to discuss more with you. At this point, I just would like to emphasize that we have different channels to stimulate the conversation between audience and speakers. We can use our Zoom channel also. We can change messages in private or public channels. Also, we have the Discord group for discussing. And I will share with you again here the website, including all posters, and then you can see again this poster if you miss some detail, and then you can contact Veronica and other tools to solve any doubt. Okay. Well, again, thank you, Veronica. And now I will move to Atama for the next speaker. Thank you so much.
Thank you.
Thanks, Alexis. So moving on.